So I want to update you on some of the research we're doing with water hemp, and I've talked about this in previous years. We're mainly doing tissue culture work, and that's because we can do genetic experimentation in a weed like water hemp without having to worry about producing seed and pollen. And so one of the things that as a long-term goal is to work in genetic biocontrol. So this is just kind of a very brief illustration here of, herbicides will target the protein, typically the site of action. There is other methods of genetic biocontrol working on RNA interference, so that is at the level of the messenger RNA. And then what we're kind of working towards would be gene drive type technology, but we're a long way from that. So we're doing some of the basics, and that's where you're actually targeting the gene. So in this case, a herbicide resistance gene, for example, with perhaps being able to reverse resistance back to susceptibility. But there's some other reasons why we're interested in tissue culture, which I'll mention as we go along. And so this is just an example of outlining kind of a long-term, a bit idealistic goal of reverting a resistance gene to susceptibility and reducing the frequency of resistance in the field. And the way one of these systems work would be to use gene editing technology. And the protein that does the work for that is this Cas9 protein. And then that will cut the gene and basically inactivate it. And that gene is targeted by using an RNA, so a nucleic acid, kind of like DNA, but single-stranded, that recognizes, in this case, the resistance gene, and then you are cutting that gene. And this system then would be in the engineered into the plant, and here you have the targeting to the resistance gene and then cutting it. And then what happens there is a little different in plants compared to some of the other organisms like mosquitoes, where this technology is being researched. And so typically a gene editing to actually reverse or to change the genetic code back to susceptibility would require a template. Once this gene is cut by the system, then the cell wants to repair that cut. And so you would give it the susceptible template, and then that would reverse the resistance to susceptibility. So the overall system is kind of like remodeling a house. You got to tear down what you got first. And that's basically the system used to cut the gene and destroy it. And then you'd give the plans to the cell. This is now how I want you to repair that. And so it would repair it back to susceptibility. Plants don't like to do that. They don't like to follow instructions like that. They rather just take the cut gene and stick the ends back together again, ignore any type of template, and do this. So here's your cut. Here's what you would like them to, the plant cell to repair to, but instead they just put the cut ends back together again. Now the benefit of that is that you do get the non-functional gene, because usually when it repairs that, you're missing some of the genetic data there. So it's not complete, so typically be non-functional. So there are ways and proposals to use that limitation in plants. And so there are a couple, and I'm not going to go into details of these. Toxin antidote system out of China, and then the Cleave and Rescue system, which is out of Caltech. Both systems to use this repair mechanism of plants to just stick things back together, but inactivate the gene, to use this in a system. And I prefer the one out of Caltech, the Cleave and Rescue, but again, I won't talk too much about it. But we are basically doing the basic research to kind of work towards testing something like this. All right, so the way the Cleave and Rescue system in general, just the basics works, you would have a functional version of that target gene that you're going to destroy, but it's different enough that it's not going to be affected by the system, so it's not going to be cut. So when you cut your target gene, in this case the resistance gene, you would have a susceptible version, but different enough that it would allow the plant to survive, but as a susceptible version and not be cut. So you don't worry about, it's actually good in that case. You're destroying the resistance gene, but you're allowing the plant to still survive so that the system can spread. That's the drive part of the gene drive, spread throughout the population. So some of the critical research then would be to choose a gene to target, and we're working with ALS, so that would be the target site of group 2 herbicides, and I don't know that that would necessarily be the one that would go to the field, but it's a gene that we know confers resistance in water hemp, and we know a lot about the gene, so it's a good one to start the work on. And so the targeting of the cutting to that specific gene is an important thing to research. And so that's why we got started with the tissue culture, is to find a system where we can work on this targeting and see how efficient it is in water hemp and so forth. So a little bit about just some basic kind of illustrative or proof of principle work in yeast, because yeast also has ALS gene. It is susceptible, so what we did was actually use gene editing to change that susceptibility gene to a resistant form, along with our own marker in there, so we know what changes we've made versus the wild type typical yeast, and then we reverse that resistance back to susceptibility again. And so on the left, we've got there are different amino acid positions in ALS that will confer resistance. One of the common one, including water hemp, would be at position 574, but it would be tryptophan, would give you the susceptible ALS, and leucine on the right give you the resistant. And so in yeast, my graduate student, Peter Bierbauer, did this work. And so first, let's talk through this one on the bottom. We've just got media that does not have yeast in it. So that's our control, no growth. So the others have yeast, and up at the top line, we've got the wild type susceptible yeast. So that's what we started out with. And then different dose response to chlorsulfuron, which is an inhibitor of ALS, so a group 2 herbicide. And from 0 on up to 300 micrograms per liter, you do see a reduction in growth. And basically at 300, you see the little spot there, but that's basically just the yeast that was added in order to begin the growth. And so you really don't have much growth there at all. And then we edited that to resistance, and now you can see much more growth at the higher level. That's probably the dose it's most easiest to see. So that would be the resistance gene, ALS resistant, group 2 resistant for a yeast. And then edited that back to susceptibility as a proof of concept of the powerful nature of gene editing. So back to susceptibility again. But again, in yeast, they will use the template that you give it and actually change the gene, in this case from resistance back to susceptibility. Plants don't like to do it that way. All right, so now I'm going to switch and talk about development of the tissue culture system, and then we'll talk about what we've been doing in terms of targeting ALS as a goal to what we're working towards. So we started out with water hemp seedlings, sterilized the seed, grew that on media, and used cuttings of the stem or the hypocotyl, and then you put that on growth media with hormones and such, just to get this undifferentiated, what's called callus, to grow. So this is just undifferentiated cell growth, but it's a good material to work with. That's not going to generate a plant or anything. You can, at least not under these conditions, but you can continue to grow it and use those cells for research. Then we took that callus culture, so the clumps of the cells, and we wanted to really break them up more, get individual cells. That way we can do like a herbicide dose response on those cells, and they're in the liquid media. It's just an easier way to expose the cells to herbicides or do transformation or something like that, put in genes rather than working with the clumps of cells. And so take those cells and put them into liquid culture, and then through a lot of propagation and a lot of subculturing, go from here, clumps of cells, more to individual cells here. But they still have the cell walls, and that will be an important point here coming up. So these individual cells, they still have the cell wall, and that can be a barrier to some of the research. So when we're looking at a gene editing system and its ability to target in this case, the ALS gene, that cell wall can be a barrier. So one of the ways that plant researchers get around that is to remove the cell walls. And those cells without the cell walls, those are called protoplasts. Right, so we use enzymes, and these are basically enzymes from plant pathogens, because when pathogens infect the plants, they'll break down the cell walls. And so that's where these enzymes come from. And we were able to get, using, I think it was the 6th or so combination of enzymes, finally worked on water hemp. And we were able to get protoplasts. And then we started to try to do transformation, but the protoplasts originally weren't really cooperating. So we were able to target the ALS gene in the protoplasts and cut, but now we want the cells to then put those cut ends back together again, right? Which is how a plant will normally repair that cut of the gene. They wouldn't do that. They weren't really healthy enough going through the process to produce the protoplast and remove the cell wall. Again, these enzymes are from pathogens, so they had kind of a pathogen response, and they were under stress, and so they wouldn't even try to do the repair. So that wasn't really helping us with the research. So we weren't able to, or we did achieve the targeting. We got the cutting, but not the repair. And here we did, for the cutting, we did just put in that Cas9 protein. That's the one that does the cutting. And here you can see this, we had a fluorescent version. So we knew everything got in there and we got the cutting, but they wouldn't divide, and they also wouldn't repair that cut. And so we took a look at oxidative stress. And in this case, the green is something we didn't want, because this is a fluorescent label that is indicating oxidative stress, and all the protoplasts were lighting up like crazy. So that was the likely reason why they weren't behaving. So then my graduate student, Peter Bierbauer, looked at different research and actually found this AIP, 2-aminoindane 2-phosphonic acid, which is being used in tissue culture actually for cannabis in Canada. So we tried it for water hemp, and it actually worked fairly well. So we were able to get then cell division, where the protoplasts are now healthy enough to divide. And here we see more divisions. Now we haven't gotten division past a few cell divisions, and that's something we're going to work on, but it was definitely progress. And so then we wanted to be able to make sure that these cells were healthy enough to actually express a transgene. So typical transformation to get genetically modified organisms and such, you need to put in your gene of interest and make sure that it's being expressed. And so that's then what we did. Now, originally, before we used the AIP, we couldn't get the cells to express. So we would put the DNA into the protoplasts. So here we've got the DNA in the protoplast. We would want, now this gene is a marker, so it will produce a green fluorescent protein. So the cells should fluoresce green. In this case, the green is what we want, but we were getting no transgene expression. But with the AIP treatment, here we can see protoplasts and about 3 of them there or so. And it's not 100%, of course, so about 1 of those then got the transgene, the marker gene, and is now fluorescing. So we know that these protoplasts are now healthy enough to both divide and to fluoresce. The other thing we did, and there's a reason for this as well, to actually use the cells with the cell wall still on. You can do transformation there using agrobacterium. So we did that as well. So that would be the number 2 here. And this is very new information. We've got that system to work. But I want you to show, or to see here, see all those clumps of cells? Protoplasts are a lot nicer to work with than these clumps when you still have the cell walls there, but we are getting transformation there. So that's pretty new information. And so the next steps then we're going to go back to with the AIP treatment to evaluate targeting and hopefully repair of the ALS gene, but into a non-functional version, and then continue to work on being able to get better cell division past just a few divisions. And so I'm probably about out of time. So you can always catch me if you have any questions.